All right, hello, this is Matt Dean with A+ College Ready, and I want to welcome you to the Week 5 AP Bio Content Review session. Please mute your microphone and turn off your webcam during the session. Make sure you feel free to unmute to ask questions. We encourage you to ask questions if you have them. We are recording this session, and it'll be posted to the A+ College Ready Science YouTube channel. Next week's session, Week 6 AP Bio Content Review, will be at 10 o'clock on Thursday, October 1st. All right, today we're going to talk about cell transport mechanisms, how things move in and out of a cell. First of all, small and non-polar uncharged molecules and uncharged particles can diffuse across the wet phospholipid bilayer pretty easily. Ions, polar molecules, and larger molecules have a harder time moving through the phospholipid portion of the membrane due to both their size or their charge. They typically have to move in and out of the membrane via either a channel or a carrier protein. Today, we're going to talk mostly about passive transport. Passive transport are types of transport that don't require the cell to use any of their own metabolic energy. Cells don't have to use their own ATP to power the movement. Usually, these processes are powered by the energy from heat in the environment. The types of passive transport that we'll talk about include diffusion, osmosis, and also facilitated diffusion. We'll start with a discussion of diffusion. So diffusion is when materials move from areas of high concentration of that material to areas of low concentration of that material until equilibrium is achieved. Once equilibrium is achieved, though, the particles don't stop moving. They continue to move, but there's no more net diffusion. That means essentially the same amount of particles move one way that move the other way. Equilibrium is maintained. Simple diffusion is how the majority of materials, nutrients, and waste are transported in cells. It doesn't require the cell to use any metabolic energy. It saves energy for things like homeostasis, repair, reproduction. Here's a quick diagram of the cell membrane. Remember that the main part of the cell membrane is composed of a phospholipid bilayer. We see that here. Phospholipids, remember, have a polar phosphate head. This side is charged, and two nonpolar fatty acid tails. And they're non-charged, non-polar. So when these substances are put in water, they form these bilayers where the fatty acid tails can point toward each other and away from water, and the heads can point toward water, which is located outside the cell, but also inside the cell. So even though we say the phospholipid bilayer and phospholipids in general are bipolar, most of the phospholipid is composed of these tails, and this part is non-polar. So this is the reason that charged particles have a hard time getting through, because they have to penetrate this non-polar phospholipid bilayer. All right, osmosis is another type of diffusion. It's a specific type of diffusion. It's the diffusion of water. Osmosis moves water, like any type of diffusion, from an area of high concentration, in this case, an area of high water concentration, to an area of low water concentration. What gets confusing about osmosis is that oftentimes we also define it in terms of the solutes, the materials that are dissolved in the water. In terms of solutes, water moves from areas of low solute concentration to areas of high solute concentration. What you've got to remember, though, and understand is if there's low solute concentration, that means that the solution's mostly water, so high water concentration. If an area has a high solute concentration, that means the concentration of water in that solution's pretty low. So really, this definition and this definition up here say exactly the same thing. This one is in terms of the water, and this one is in terms of the solutes. Osmosis can also be defined, though, as movement of water from areas of low osmolarity to areas of high osmolarity. That essentially means low solute concentration to high solute concentration. So osmosis tends to happen in cells when there's a membrane that's impermeable to a particular solute, but that membrane is permeable to water. So since the solute can't move, water moves to even out the concentrations on each side of the membrane. So that's an important thing to keep in mind. Osmosis tends to happen when there's a solute on either side of the membrane that can't move through the cell membrane. Therefore, water moves to even out concentrations. Most water doesn't go directly through the phospholipid bilayer. Most water actually flows through these specific protein channels for water called aquaporins. And these proteins essentially act like tunnels for water movement. It's a type of facilitated diffusion. Osmosis is crucial for all cells. Osmoregulation is the process of regulating the solute and water concentrations of the cells and of the body. It's one of the main functions of the urinary system, and it's crucial for life. Some terms that you need to be familiar with. If you say that a cell is turgid, this refers to a condition when there's lots of water in a cell. So the cell becomes rigid and stiff. Think about it like a freshly cut piece of potato. Flaccid is when there's not a lot of water in the cell and it becomes limp and wilted. Think of that like a plant that just hasn't had enough water. It's been on a hot, sunny day. Plasmolysis is when a cell membrane shrivels away from the cell wall. This happens in cells with walls, obviously, like plants. And it happens when a cell is put into a hypertonic solution. So typically, if we had a cell with a wall, the membrane is going to be out here against the wall. But if you put a cell in a very salty hypertonic solution, and by the way, hypertonic means more concentrated than what you're comparing it to, water is going to move out of that cell, and the cell itself is going to shrivel. Now, the wall doesn't shrivel because it's a solid, hard substance, but everything inside that membrane shrivels away from the membrane so that the cell may look more like that instead of that. Since we mentioned hypertonic essentially means the solution is more concentrated in terms of solutes than what we're comparing it to. The opposite of that is hypotonic. That's a solution that has less solutes in it than what we're comparing it to. And we might also refer to a term isotonic. So this refers to two solutions that have the same solute concentrations. Yet another way to define osmosis. Osmosis is the movement of water from hypotonic areas to hypertonic areas. Areas with lower solute concentrations and higher water concentrations to areas of higher solute concentrations and lower water concentrations. So a couple more terms. The ability of an extracellular solution to make water move into or out of a cell by osmosis is called tonicity. That's a word we'll see a lot in AP biology. And then another important term is osmolarity, which is essentially the total concentration of all solutes in a solution. Both those are really important terms for this unit. So a solution with low osmolarity, low solute concentration, has fewer solute particles per liter of solution, while a solution with high osmolarity has more solutes per liter of solution. When solutions with different osmolarities are separated by a membrane that's permeable to water but not to the solute, water will always move from the side with the lower osmolarity, the lower solute concentration, to the side with the higher osmolarity, the side with the higher solute concentration. All right, another important concept in this unit is water potential. This is one of the math concepts that are tested on the AP exam fairly regularly. So we're going to talk a little bit about water potential. Water potential is often abbreviated or represented with the Greek letter psi. Sort of looks like Poseidon's trident. Poseidon's the god of water, so that makes sense. Water potential is a measure of water's potential to move. In terms of water potential, water always moves from areas of high water potential to areas of low water potential. Water potential is made up of two important parts: the pressure potential, which is abbreviated psi sub P, and the solute potential, which is abbreviated psi sub S. Those two together add up to give you what's called the total water potential, which you'll see abbreviated as either psi or sometimes psi sub T, total water potential. So if we only accounted for solutes, we could deduce that water would move into the plant cell in the diagram continuously, because in this beaker, there's distilled water, which means there's no solutes. A plant cell has lots of solutes in it. It has sugars and salts and lots of other things. So water always moves from areas of low solute concentration to areas of higher solute concentration. So water's going to move in. And we would think that it would always move in. Assuming that the solutes can't move out, water would just keep moving in forever and forever and forever. But we know that doesn't happen, because eventually, as more and more water moves into this cell, the pressure inside that cell starts to build up, and that pressure is pushing outward. Eventually, so much pressure builds up inside that cell that even though the solutes cause water to want to go in, it can't, because pressure is pushing out too much. So water potential takes into account both the effects of the solutes and of the pressure. Our more generic version of osmosis is movement of water from areas of low solute concentration to areas of high solute concentration doesn't take into the account the effects of pressure, which are very, very important for cells that have cell walls, cells like plant cells, fungal cells, bacterial cells. So again, the value of looking at water potential is that it takes into account the effects of pressure instead of just looking at the concentrations of the solutes. Some important things to know about water potential. The water potential of pure water in an open container is set to a value of 0. This is because there's no solutes, and the only pressure acting is atmospheric pressure. The pressure potential of the system is typically 0. Unless you're provided a pressure value in a problem. So most problems you'll see in AP Bio are going to involve 0 pressure. Every now and then they'll throw a pressure value at them, and we'll see one of those in a problem in a minute. As more and more water enters a cell, the pressure inside that cell increases. We also have a formula on the AP Biology formula sheet to calculate solute potential. And we can see that formula right over here. So the solute potential is equal to negative I CRT, where I is called an ionization constant. And it's really important, especially if we're talking about salts. First of all, if we're talking about sugars, anytime you have a solution that has sugar as its solute, I for sugars is always equal to 1. Because if you dissolve a sugar molecule, one sugar molecule doesn't break into pieces, it just dissolves. So one sugar molecule as a solute is still one sugar molecule in solution. On the other hand, if you have a salt, say like sodium chloride, if you start off with 1 formula unit of salt, not dissolved yet, and you put it into water, that 1 formula unit of sodium chloride breaks into 2 ions. So I for that solution is 2. If on the other hand we had a solute like, let's say, calcium iodide, if we put that in water, it might ionize into calcium ions plus 2 iodide ions. That's 3 ions. So I for that salt would be 3. Important thing to remember, I for sugars is always 1, and I for salts you determine by ionizing that salt and figuring out how many ions 1 formula unit of this salt will break into. For sodium chloride, that's 2. For calcium iodide, that's 3. C is the concentration of the solute. So it's going to be the molarity of the solute. That's generally going to be given to you in the problem. R is the pressure constant, which you've probably seen in chemistry class before when you were dealing with like PV equals nRT. Now, this value is probably a little different than the one you used in chemistry for R because it has different units. Same constant, in this case, measured in different units, but 0.0831 is the value we will always use for R. That value is given to you on the AP Bio formula sheet. T is temperature. The temperature has to be in Kelvin for this to work. It's an absolute temperature. They're probably going to give you temperature in Celsius. To change from Celsius to Kelvin, you add 273. So remember that when you're given a Celsius temperature, add 273 to it to get to Kelvin. Again, all of this is included on your AP Bio formula sheet, which you can use throughout the whole exam. This is a screenshot of part of that formula sheet. So a couple of other important things to remember about water potential. The more solutes that are present, the lower the water potential. And that's because of this negative sign in the formula. Solute potential equals negative I CRT. So more solutes, lower water potential. We already talked about how to calculate the ionization constant. Always 1 for sugars because sugars don't ionize. For salts, break up the salt into ions and figure out how many ions form per formula unit of salt. C, we already said, is the molar concentration, the molarity. R is the pressure constant or gas constant, 0.0831. And T is the Kelvin or absolute temperature. That being said, let's look at some problems like you might see on an AP Bio exam. So the molar concentration of sodium chloride solution in an open container is 0.30 molarity or molar. The pressure potential of the solution is 0. What is the total water potential at a temperature of 25 degrees Celsius? So remember that water potential is the pressure potential plus the solute potential. Well, we were just told that the pressure potential right now is 0. So we need the solute potential. Our formula for solute potential is negative I CRT. So for sodium chloride, it's a salt, it's a metal and a metal. We're going to ionize that thing and simulate what would happen when it dissolves. So one of these ionizes into 2 distinct ions. So I for that solution or for that solute is 2. So we got -2. The concentration, the molarity given to us in the problem is 0.3. And it's important to remember that molarity stands for moles per liter. I'm going to write it that way to show you how the units cancel out. R is 0.0831, and that's liters times bars over moles times kelvins. And then the temperature is given to us as 25 degrees Celsius. So we need to add 273 to that. That'll give us 298 degrees Kelvin. Now notice what happens to units. Kelvin here cancels with Kelvin here. Moles here cancel with moles here. Liters here cancel with liters here. And that leaves us with a unit of bars. That's what we're going to typically measure both solute potential and water potential in. Let's go ahead and do the math on this. So let's go 298 times 0.0831 times 0.30 times 2. And remember that that's a negative number. So our solute potential equals negative 14.858. So we're going to round that to negative 14.9 bars. So that's going to go right here. That means our total water potential in the container is negative 14.9 bars. Because water potential is a pressure of 0 plus a solute potential of negative 14.9 to give us, here in this container, a total water potential of negative 14.9 bars. So we've answered the first part of the question. But if we go on down, if a plant cell with a total water potential of negative 5 bars is placed into a beaker of the solution described above, will water move into or out of the cell? Assume the cell's membrane is impermeable to the solute. Justify your answer. So we're saying inside this cell, the water potential is negative 5. Remember that water always moves from areas of higher water potential to lower water potential. The negative numbers mess people up. You've got to remember that -5 is a bigger number than -14. -5 is closer to being positive. So -5 is larger than -14. So water is going to move from higher water potential in the cell to lower water potential in the solution. Water is going to move out of the cell because the water potential in the cell at -5 is higher than the water potential in the solution at -14.9. So we could say that this cell is going to plasmolyze, because what will happen inside is that the contents of the cell will shrivel away from the wall, and it might look something like that when it's over. All right, let's go to problem 2. So same kind of setup, except this time our solution is, our solute rather, is sucrose, a sugar. We know it's a sugar because its name ends in OSE. It's important to remember that sugars don't ionize, and therefore I for solutions containing sugars is always 1. So this tells us that we have a sucrose solution in an open container. The molarity is 0.4. The pressure is 0. We're looking for the total water potential at a temperature of 27 degrees Celsius. So again, our pressure is 0. Let's calculate our solute potential using -I CRT. We already said that since this is a sugar solution, I is 1. The concentration is 0.4. And again, we're going to write that as moles per liter so we can cancel units. R is always 0.0831. And that's liters times bars over moles times K. And our temperature is 27 Celsius. We need to add 273 to that and get 300 degrees Kelvin. Cancel units. K's cancel. Moles cancel. Liters cancel. And now we do the math. So we'll go 300 times 0.0831 times 0.4 times 1. And remembering that this is a negative number because of that negative out front. So our solute potential comes out to be -9.97. That goes right here. And therefore we end up with a total water potential inside this beaker of -9.97 bars. So that's the answer to this part of the question. Now we move down and look at the bottom part of the question. If a plant cell with a total internal water potential of -7, so inside this cell, the water potential is -7, what will happen? What will happen to the cell's total internal water potential as time passes? Assume the cell's membrane is impermeable to the solute. So we're asking a different question here. First of all, we've got to think about what's going to happen to water. Is water going to move into or out of the cell? And the answer is, since -7 is a bigger number than -9.97, water is going to move out. So let's think about what that does to the total internal water potential of the cell. So as more and more water moves out, the pressure inside that cell is going to go down. And also, as more and more water moves out, the solute concentration is going to go up because the water is not diluting it anymore. So if we think about this equation, as this number goes up, the solute potential actually becomes more and more negative. It actually decreases. So in both cases, pressure is going to decrease, solute potential is going to decrease, the total internal water potential of that cell is going to decrease because the pressure drops and the solute concentration is going to increase, therefore decreasing the solute potential. All right, one more sample problem. Here's one with a solute of calcium chloride. So this is a salt because it's made of a metal connected to a nonmetal. Again, we want to calculate the total internal water potential, or the total water potential of the solution. We're given a pressure of 0. So we need to calculate the solute potential. -I CRT. For calcium chloride, when this breaks up into ions, it breaks up into a calcium ion and 2 chloride ions. So one formula unit of calcium ion ends up ionizing into 3 distinct ions. That means I for this salt is 3. So -3 times our concentration of 0.08. And remember, that's moles per liter. We're going to multiply that by R, 0.0831 liter bars over moles times K. And we're going to multiply that by the temperature, which is 37 C. Add 273 to that to get 310 K. Cancel out units. Everything, once again, cancels except bars. Now we do the math. So that's going to be 310 times 0.0831 times 0.8 times 3. And we end up with a negative number, as always. And that's going to give us a solute potential of -61.8 bars. So that's the total water potential inside here. So once again, we put that in, put a cell with a total water potential of -16 in there. Once again, this number, -16, is bigger than -61. So water is going to move out of the cell from higher water potential to lower water potential. And this cell again would plasmolyze. Now, if on the other hand, let's say the water potential inside this cell had been like -120. If that was the case, water would move into the cell. And it would move from higher water potential in the beaker to lower potential in the cell. And this cell would start to build up turgor pressure. It would become more and more turgid until eventually the pressure would get high enough that no more water could enter. All right, that's a brief review of water potential. One more thing about water potential on this slide. So here we have 2 sides of a membrane. Here's the membrane running down the middle. And we're looking at side A, has a solute potential of -1.7 and a pressure potential of 0 bars. All these little triangles represent solutes. Side B, we have a solute potential of -0.6. The triangles again represent solutes. And this time we have a pressure potential of +2 bars. So sides A and B are separated by a semi-permeable membrane. Assume that the cell's membrane is impermeable to the solute, but permeable to water. The triangles represent the solutes. If the volumes of the 2 sides are initially equal and the membrane is impermeable to the solute, in which direction will water flow? So let's say they both start off with a water level like this. We're thinking vertically here. So over on this side, the total water potential is going to be pressure potential of 2 plus solute potential of -0.6. That's going to give us 1.4 bars on this side. On this side, solute potential of -1.7 bars, pressure potential of 0 bars, giving us a total water potential over here of -1.7 bars. We know that water moves from areas of higher water potential to areas of lower water potential. So water is going to move this way. So over time, what's going to happen is that this water level on the left is going to start to go up, and the water level on the right is going to start to go down, like so. So we answered the water potential on each side. Which side is hypertonic? Remember that hypertonic means more solutes, a higher solute concentration. Well, that's side A. It has a higher solute concentration, which means side B has a lower solute concentration. The word for that is hypotonic. What will happen to the volume on side A? Well, it's going to go up because water's moving from B to A. The volume on side B is going to decrease. And then why is the initial solute potential on side A lower than the initial solute potential on side B? Well, we got to think back to that equation. Assuming everything else is the same, R is a constant, assuming that the temperature is the constant. When there's a higher concentration, that makes this number become a more negative number. And the more negative the number, the smaller the solute concentration. All right, we want to end up today thinking a little bit about transpiration and how water potential relates to that. So these are water potential values throughout a tree, essentially. So in the root, or in the soil rather, the water potential is negative 0.3, but in the xylem of the root, it's negative 0.6. Well, that tells you that water is going to move from higher water potential to lower water potential. So from the soil, water moves into the xylem because of water potential. Well, up here in the trunk, the water potential is negative 0.8. Well, the water potential in the root is higher than that. So water moves from root to trunk. Well, in the leaf, the water potential is negative 0.1. Well, in the trunk, negative 0.8 is higher than negative 0.1. So water moves from higher to lower water potential. In the air spaces around the leaf, that water potential is negative 0.7. So again, water moves from higher to lower water potential. And then finally, outside the stomates, the water potential is even lower. So water moves from inside the leaf out to the air around it. So all the way through the process of transpiration, water is moving from higher water potential down here to lower water potential up here. So water potential is one of the main driving forces that moves water from soil into plant, up to plant, and out into the water, or out into the air rather. All right, we've got a couple more types of diffusion we want to, or cell transport rather, that we want to briefly mention. One of those is facilitated diffusion. So the word facilitate means essentially help. Facilitated diffusion is a type of passive transport. Essentially, it's a type of diffusion where the solute needs help to make it through the cell membrane. And that help is provided by a channel protein, a protein that's embedded in the cell membrane. So facilitated diffusion is the transport of materials from high concentration to low concentration through a channel protein. This type of transport is typically really important for materials that are either polar, ionic, or very large, materials that otherwise couldn't move through the cell membrane. The movement of water in and out of a cell largely occurs via facilitated diffusion. Aquaporins are a type of channel protein that help water to move in and out of a cell. There are also gated ion channels that help to move things like sodium and potassium in and out of cells. These are very, very important in neurons. Active transport is another type of cell transport. Active means requires the cell to use its own metabolic energy. Essentially, this means it requires the cell to use its own ATP. This process moves materials against the concentration gradient. It's like moving things from low concentration to high concentration. Maybe the most famous example of this in the body is the sodium-potassium pump in the nervous system. This requires ATP to move sodium and potassium ions against their concentration. This is also important in photosynthesis and cell respiration, where hydrogen ions are pumped against the gradient. Bulk transport is another type of active transport, requires energy. Bulk means big. So this is the movement of molecules that are too big for proteins to transport. It's too big for these channel or carrier proteins to move. There's two main types of bulk transport we'll talk about in AP Bio. One of them is exocytosis. If we break that word apart, exo means outside, cyto means cell. So this is the process of moving large materials out of a cell. Sometimes we'll refer to it as secretion. An example of this would be like, when pancreatic cells release the hormone insulin into the bloodstream and the cells and the fluids around the pancreatic cells. Typically, this involves the vesicles from the Golgi apparatus filled with insulin, fusing with the cell membrane, and then pushing insulin out of the cell and into the interstitial fluids that surround it. Endocytosis is essentially the opposite of exocytosis. This is bringing large materials into a cell. We really break endocytosis into two parts. Phagocytosis, which is bringing in large solid materials into a cell. This usually involves surrounding the particles with the cell membrane, engulfing those particles, surrounding them with a vesicle, and then using structures like the lysosomes to break down and digest those materials. A good example of this would be a white blood cell phagocytizing a bacterial cell, taking it in, and then following that up with intracellular digestion carried out by the lysosomes. Pinocytosis is essentially bringing in dissolved substances or liquids via the process of invagination and endocytosis. This might be bringing in certain kinds of hormones, certain kinds of needed solutes in large amounts into the cell via the process of surrounding and engulfing those particles. Oftentimes, this involves those solutes binding to receptors, which then stimulates the pinocytosis of those materials. Here we can see these things happening. So here's phagocytosis of a large solid particle. We can see it's engulfed and surrounded in a vesicle. Here we see pinocytosis happening essentially the same way with dissolved substances. And then here's the receptor stimulated or receptor mediated version of pinocytosis. First of all, those things bind to receptors that triggers the endocytosis of those materials. All of these are forms of active transport because they require the use of ATP by the cell to move the particles. Here we can see exocytosis happening. So the Golgi apparatus has bundled up and transported a substance to the cell membrane. That vesicle bumps into and fuses with the cell membrane. The vesicle itself, since it's made of phospholipids, becomes part of the cell membrane, but the materials inside, whether they be proteins or other materials, are pushed out of the cell. That's secretion or exocytosis. All right, we'll end today by talking about the last type of cell transport that we're going to look at in AP Bio, and that is co-transport. So co-transport is a process in which two substances are simultaneously transported across a membrane by one protein. Usually this protein doesn't require ATP. It doesn't have any ATP-ACE activity. It is a type of active transport, though, even though ATP is not being used. Typically, the energy from transport of one material is used to power the transport of the other material. So usually one substance is being moved with the concentration gradient, high to low concentration. The movement of this substance provides the energy to transport the other substance against the gradient from low to high concentration. So an example of this is the absorption of glucose by cells, the epithelial cells in the gut, essentially in the small intestine. In that case, glucose is co-transported with sodium ions. The concentration of sodium ions is higher outside the gut cells than it is inside. We can see that out here. Lots of sodium on the outside, not so much on the inside. Sodium moves into those cells down the concentration gradient. So sodium's going to move from higher to lower concentration through this protein right here. This into a cell. So essentially co-transport is when the movement of substance with a gradient provides the energy to move the other substance against a gradient. All right, that's it for our methods of cell transport. I hope that helped, and I hope to see you guys next week, October 1st at 10 a.m. for our week 6 AP Bio content review session.